Okay, good afternoon, everybody. Thanks for joining us for the Digital Ag Seminar Series, midday Thursday. Nice attendance today. Today we're lucky to have Chris Topp join us on campus for a day and a half or so, and have a chance to engage the broader plant science community and others, actually. And we're excited that Chris is here because he's really recognized as a distinguished expert in root biology. We have a lot of people here on campus that are studying roots. We have varying degrees of expertise, and so it's exciting to have Chris here to join us to chat a little bit about his work and to hear about potential opportunities to engage. And so he's going to tell us some of the work that he's doing in his lab. Thanks for joining us, Chris.
Oh, I'm so happy to be here.
And I should say he's from the Danforth Center.
Oh yeah, just down the way. Yeah, I drove here last night. It was a great drive. Got to see what's happening with the corn and soybeans these days, which is always useful, at least the above ground parts, right? So today we'll be focusing on what's happening below ground and some of the tools and
Is that acceptable?
Yeah, technologies that we use to get there. You know, one of them being, maybe you deduced this, we involve a lot of X-ray imaging in the lab, and I'll show you a few examples of that. But I want to touch on several things today, including some of the genetics that we've learned, including some of the more mechanistic discoveries, and also where we're moving next in terms of thinking about cropping systems and some moving some of our discoveries into the applied realm. Okay, so yeah, so the lab basically has three kind of paths. So one is developing these phenomics tools that allow us to study roots and therefore allow us to study the whole plant. So what you're seeing here is a positron emission tomography data set of 5 minutes of photo assimilate moving from leaf to root system. So like tracking in real time how the plant is allocating its carbon. And then once we have better tools, the idea is that'll give us better insight into genetic and mechanistic basis of root growth and root environmental interactions. And then the third part is more recent. In the last 3 or 4 years, we've had some success with 1 and 2. And so it's now how can we use these tools and our learnings to really move towards more sustainable agriculture goals. So that's it in a nutshell. Okay, so why do we choose to study roots? I mean, it's a hard question, and basically there's so little known about it. It just seems like an obvious black box to shed some light on, right? And the argument's not hard to make for roots. They're super important. They're literally the foundation of what we see above ground. And when we start to think about soil health, interactions with the soil, it's really roots, at least from my plant-centric perspective, that are driving the rhizosphere, which results in a lot of chemical, biological, and physical change to soil that we're interested in. And thinking about it from the perspective of just crop improvement and sustainable ag solutions, basically my group in some form or fashion touches on all of these different areas because they all have fundamentally start at the root soil interface. So some of these I'll touch on today, some I won't. But anyways, that's why roots. Okay. So this is a concept of root system architecture, which is, can be stated pretty simply. It's the spatial and temporal organization of root systems. And we think that root system architecture, like where and when roots are, matters for efficient nutrient capture. Here's a really simplistic diagram, showing a difference between maybe a shallow rooted and deep rooted plant earlier in the season. And the ability to capture phosphorus and investing roots in the topsoil is pretty well established at this point. A more relevant problem to a lot of our Midwestern agriculture is, for example, early season drought, and obviously plants that invest in deep roots have an advantage there. We actually had one at our farm this year, and so we've got to evaluate some of our deep-rooted genetics and how they did, this year. So this is quite relevant, I think. And then, of course, over the course of the growth season, the water issue is still a concern. But when you think about, again, very basically nitrogen dynamics, and that nitrogen, is moving basically from the soil surface where it's applied down through the soil column. And so plants with deeper roots and that have deeper roots earlier in the season basically have more contact with that end and theoretically should have enhanced ability to capture nitrogen either more efficiently or just to capture just more in general. Okay, so it's an idea, and what we'd like to be able to do is test this out, and it's hard to test some of this stuff out because we don't have the ability to manipulate roots. So that's largely what my program has been focused on. There's another aspect which we're pretty and keen to think about in the last 3 or 4 years, especially as we have the ability to manipulate root systems, thinking about soil carbon sequestration, and thinking about putting deeper roots or more root mass deeper into the soil and how that might stabilize carbon and sequester it for longer periods of time deep in the soil, right? There's been a lot of modeling work around this. Keith Paushians, for example, has modeled some work for DOE, and we've been funded on a few DOE projects in this area. And the number gets quite big if we can increase root biomass and move it deeper in the soil, at least theoretically, in terms of the amount of carbon sequestration that can happen. I mean, it's on a real scale of like gigatons per year, which is starting to be relevant to like large scale carbon sequestration. But this is basically largely untested because it takes a long time to do this work, like many years, 3, 4, 5, 10, I don't know, if we're learning this. And it's really hard because it's hard to measure roots, especially 50 centimeters or deeper in the soil. Okay, so yeah, hard to do. Fact of the matter is, that's my team 2 years ago, taking root exudates from these corn plants. We dug a big trench. This is what Weaver and Kucher and Kesselbach did, basically 100 years ago, dig a trench and do some archaeology. This is is it overstating it? I mean, we do have new tools in the field, but there's really not a magic root phenotyping system that we can just go in and sweep over the field like a drone or something and capture like a high resolution root data. It just doesn't exist. And, I hate to say it, but I don't think it will exist anytime in the near future. I would not hold your breath for it. Okay, so we do develop, this is apparently on a timer, so I'm just going to try to keep pace with what the PowerPoint tells me to. So that's probably useful in terms of finishing on time. So we have a number of tools in the lab, and I'll touch on a few today. One we're pretty well known for is X-ray imaging. We acquired this system on the left, which is an industrial X-ray CT. It's made for like aerospace, like factory shop floors. And we got this in 2016. The advantages of it are it's got a really big cabinet, so we can put big things like plants in. And obviously, unlike a medical scanner where the detector and the source go around in that big donut thing to a patient lying horizontally, this has got a 90-degree orientation. So basically, we spin the sample instead of the detector and the source, and we can basically image over, basically a 2 by 4 foot region. I mean, that would be an enormous scheme. We don't really do that very often, but theoretically, we could in the system. And a few years later, we acquired another system which actually includes microscopy objectives, and that allows us to get to sub-micron resolution and to see like really small things like the structure of soil aggregates, or as I'll show you, so like microbes, like on roots and in soil. Okay, so these two systems together are a really nice complement for us. And if you're on Twitter or X or whatever, Keith he runs the systems. I literally hired him on the day we got this system, and he's really makes them sing, and he has a lot of domain knowledge in the area. And he's a good follow on Twitter because lots of pretty pictures. But he's a good person to talk to as you develop your own X-ray tools. Okay. Oh, and if you want even like really amazing data that comes out of the X-ray microscope, check out this paper in Plant Phys a few years ago, or if you just want to look at the pretty videos and pictures, just go to the Figshare link, and there's a bunch of really neat stuff to look at there. But I'll share a few of these with you. So again, we focus on roots. So this idea of like kind of doing multi-scale analysis, this is a sorghum root crown, X-ray scanned and then rendered, to look kind of rooty looking. But then we can take pieces of this and scan them in the X-ray microscope. So here you're looking at, basically we're able to, count tissues and cells here in a root tip, and here look at the nodal plexus where the nodal roots form to really understand development. There's a lot of applications for vasculature, which are like sort of obvious, but just outside of really our current program. I would like to do some work in this area because just the data is incredible. We just don't have the bandwidth for it. So open to entertaining ideas in that area, among others, for sure. Then, of course, making these pictures is these days like the easy part. The hard part is now turning what we see naturally with our meat computers, our brains, like pattern recognition, into data that we can actually do science on. So this is this image processing and segmentation steps, right? And so we collaborate widely with computer scientists and engineers to develop, and I think this group understands this well, obviously, with your phenomics efforts, but this is often much more than half the battle. So we can count cells on a root tip. Here we're looking at a piece of root that's been infected with a vascular mycorrhizal fungi. You can see the root hairs, and then in yellow here we've identified what are like basically, these probably are not arbuscules anymore. These are like vesicles that are left over after the AMF has kind of done its thing. But the idea is we can start to digitally dissect the different components of this system at a fine scale, and then also understand it in the context of this is not a real soil, but like a reconstituted artificial soil structure, right? So in this case, we were interested in what was inside the root. Here's another example of seeing these AMF. You can see some of the hyphae here, and these little balls are the spores. So these are outside of the root. We haven't quite labeled the root in this data set, but it's here. You can see some of the stuff that's in the root and some that's out. So we can really sort of understand the spatial context, like in terms of soil pores and soil particles, as well as what's inside and outside the root with some of these tools. So that gives us some of that really fine-grained structural information. And then, I mean, even though we're a root lab, actually the easy stuff is like kind of also above ground. So we've done some work here just because it's quite effective, and interested in plant development in general. So this is, this is basically the inflorescence meristem. So this is what, these stem cells that give rise to the flower, everything we see above ground. This is in the Subterranean model grass system. And so scanning this and doing segmentation, we can see each of the branch meristem. They're all different. They're just random colored, but each one is like its own set of branch meristems that then go on to become the flowers and the seeds of the plant. So it's really quite effective at that scale. And that allows us to get really early in the process. But maybe a more practical approach for, sort of the more agronomy side is measuring complex structures like a sorghum seed head and the variation in that. This is very difficult to do by hand. I mean, you can pick all the seeds off, you can get a ruler, you can use 2D image analysis, but it's really fundamentally a three-dimensional shape that's quite complex. And so, we effectively have, I think, been able to use the X-ray effectively to get like high information content phenotypes from sorghum. So we published a paper a couple of years ago with like 100 of these. We tried to survey across the 5 major botanical races of sorghum and ask questions about what kind of phenotypic similarity or diversity was there, and was there evidence there's evidence of genetic separation in these races, but is there evidence of phenotypic separation? The answer actually was largely we did not see it. This is a small subset, and this is not the final word on it, but it does provide some new insight just because of the amount of phenotypes we're able to measure, right? So essentially, we can take a sorghum seed head, and we can measure all the seeds, where they are, the size, the shape of those seeds. We can further dissect into the branches, the nodes on the stem, et cetera, et cetera. So we can really dissect this and measure everything that we think is elemental to building that complex shape. Okay. So that's all I'm going to say, I think, about above ground stuff. So getting back to the roots, of course, we have the exact same problem, even if the roots maybe in some sense aren't as, don't look like they have as many features. By eye, we can see differences in these, right? These are different inbred maize lines from the NAM founder panel from around the world, mostly around the country. North Carolina, Ohio, Texas, Missouri. These are from Summit, et cetera, right? And we can see the shape, which is probably to some extent due to the adaptation to where they were bred. But we need to turn this into data, quantitative measures, so we can understand genetics, right? For example, that's at least my interest. And so it's the same idea, with these roots. So we dig these up by the thousands. This is just the root crown. There's obviously all the roots below in the soil we're not getting, but the root crown is the nexus of all the roots. So all the roots in the root system are connected here, essentially, right? They're all connected here. So we get the beginnings of each of the entire root system. So it's pretty information rich, and obviously there's variation. So there's definitely genetic signals. I'll give you an example of that. And so, this is a bread and butter thing we do in the lab. It's one of the simpler uses actually of an X-ray system, because we're not segmenting soil, we're not scanning through a pot, we're just scanning this lignified root crown, which basically holds its structure pretty well in place. So we can do this through root crown pulling or shovel excavation, and we have a whole pipeline we developed that basically captures basically 100 traits, right? And these traits are, some of them are kind of obvious, like total root length or number of root tips or angles of roots. Some of them are a little more esoteric, like the distribution of biomass in the 3D realm. And we basically use statistics and statistical genetics to sort out which of these different ways of measuring this complex shape give us a biological signal and are therefore useful in some form or fashion to our science. Okay, so that's sort of our approach. This is a little bit different than the approach where we're trying to identify what are the core phenotypes that sort of build the emergent properties eventually. That's another way of doing it. We tend to be a more data driven. We measure as much as we can about a complex shape, and then we use statistics to tell us what the signal is, so we're not presupposing what's important and what's not about the root. That's the approach we take. So again, if you're interested in this sort of work, we've built on it and continue to build. We just literally submitted this Topa Root Plus yesterday, but this goes even further than what I showed you before, and really allows us, just like the sorghum example, to break down. So we were measuring, which nodal roots are connected to which nodes, how many lateral roots are on each nodal root. I mean, literally, if you could just pick the thing apart but keep the 3D information in place, that's what Topper Root does. And then the new one has some advanced features and the proto-GUI and things that are a little easier to use. Okay, but of course, the hard part, the thing I really got into the X-ray game for, was to measure roots growing in situ, this isn't soil, it's an artificial clay substrate. But that's the idea, growing in some substrate and eventually reconstitute field soils is the goal. But we've done most of our work in simpler systems so that we could make faster progress. Like many plant phenotyping and computer vision approaches these days, we really found a lot of utility for artificial intelligence and UNets. Neural network has worked really well for image-based data. It worked really well for us. In this case, so obviously you're looking at a soybean plant, and then these little colored things are the different nodules on it. And this now allows us, the other thing I didn't mention there, because the slides are advancing on their own faster than I'm explaining it, sorry about that, but is we maintain the topological integrity. So there aren't gaps in the root system. This isn't something that a UNet does natively, but we did some refinement on using the probability maps that are output. And so basically the roots are all connected. Okay, so it's one topological unit, and I got to go backwards. This is really, I don't know how, it's ghost in the machine, but this is important because that allows us then to take time series of this where we feel pretty confident we have all the roots and they're all connected, and lay them over one another. And so we use this thing called the time function, which allows us to basically measure new roots that form and how much each individual root grew at a later time point. And you might see that some roots grew a little and some roots grew a lot. So there's a lot of heterogeneity in this. And this is also reflected in these scatter plots. So here you're looking at growth rate of root length, and here's a 3D version, which is just root volume. It's kind of like biomass. And I mean, there's a correlation, but it's not a very strong one, right? So generally the largest roots will grow faster, but actually even in a seedling root system, this is probably a 3-week-old soybean plant. There's a huge amount of heterogeneity, and so this is probably very important as we think about root growth simulation models, right, which tend to produce very uniform roots, and so we're not quite there yet on those, I think, and those are really valuable for hypothesis testing, and also just how roots are interacting with the environment, how that feeds back into their growth and development. It's really not this even thing. These micro scale interactions at each root tip are, I think, greatly affecting how that root grows, and over time, the emergent property of the 3D architecture is what we get from all of these microprocesses. So it's really important to like understand what's happening at that local scale, iterated over hundreds, thousands, tens of thousands of roots. That's what gives us the root system architecture, which is the functional like thing where we think about in terms of nitrogen uptake efficiency or drought tolerance. In my opinion, that's my equation. This. So I'm going to move now into some genetics. So we've got all these phenotyping tools, and so now we wanted to use them to identify genes or gene variants so that we can, like build a better root system, right? In some cases, we want to build a deep root system, some cases a shallow one, in some case one with few long laterals, in some cases one with many short lateral, whatever it is, right? We want a toolbox that allows us to like design the root system for the environment, but we just don't have any of those tuning knobs. So that's what we're looking for here. So there's this well-known example, steep, cheap, and deep, from Jonathan Lynch, who's like the godfather of like root system architecture, basically. And he's proposed a lot of ideas, and some of them are empirically validated, but many of them have really yet to be tested, especially in across a lot of systems and environments. So this idea of steep, cheap, and deep, there's aspects of it. The cheap part has to do with the metabolic costs of growing things. I'm not going to get into that today. We don't quite study that so much in my group, but thinking about steep and deep gets to that efficient nitrogen capture, or drought avoidance, or drought tolerance, however you want to think about it, in terms of making root systems basically narrow and deep and getting in there quickly. So one of the ideas is this one rule of high occupancy, so like many nodal roots near the soil. And the idea of that is you have many more roots, and because this sheet is an axis of time, these are the youngest nodes. So these roots have had the longest time to grow. These ones form later and later and later. So the higher you get, the less time they've had to grow deep. And also they start from a physically higher position to the soil. Many of them never reach the soil. The ones that do are obviously start shallow, and they don't have as much time to grow deep unless they're growing really fast, which they could be. But this is a model. So the idea is if you have a lot of these nodal roots kind of right here or just right above the soil, you'll get lots of roots, you'll get them deeper, you'll get them there faster, you can capture more nitrogen that way because there's just more touch time, more touch points, more touch time at the end. So we've got a gene that we think helps with this. We've also been working with the deeper rooting genes, which are well known for drought avoidance in rice, and have homologs in basically all the sequence genomes we've ever looked at. They do a very similar thing in corn as they do in rice. Arabidopsis, they basically affect gravitropism at root tips. And so like if they're overexpressed, they make really gravitropic deep roots. If you knock them out in different combinations, because there's 3 in maize, they'll make progressively shallower root systems. So these can definitely affect that. That work's a little less advanced, a little more complicated, so I'm not going to talk about it today, but hopefully I'll also get to another gene we found through genome-wide association phenomics approach, which is we call just ideal root architecture. That name probably won't stick, but it's stuck for like 5 years now because I keep saying it in seminars. But anyways, we needed some name for it. I don't know if ideal, that's a little idealistic probably, but pretty good root architecture one might be like a better way to describe it. We found this through a GWAS, and it's involved in nitrogen responsiveness. So I'll tell you a little bit about that story as well. And so these things kind of match the steep, cheap, and deep idiotype, or allow us to test some aspects of it, I guess is the idea. So I'll start with rootless. This is a really cool, I'm a maize genetics person, born and raised basically the last, 25 years now. I've worked in maize systems. And so rootless 1 is this classic maize mutant. It was identified in 1930 in a farmer's field. The corn kept falling down, and actually it could have been in Indiana. I should know this being in this audience, but it might have been found in Indiana. But it was brought to the local university, maybe Purdue, I don't know, it could be. And basically to Jenkins, right? Where did Jenkins come from? Does anyone remember back in 1930? No. So, but it was described there in a paper, and it has been maintained in the genetics stock center in the germplasm since then. So something to keep in mind, this is obviously like an open pollinated variety. It was something being grown in 1930. This is not a modern hybrid or modern inbred. It was before inbreds and hybrids existed, basically, like right on the edge of it. Okay, so no nodal roots, but if you stake it up, it'll make seeds and you can propagate it. So this isn't very useful in and of itself, but I'll get to why I think it can be useful. And so this has been, the mutant was there. We knew it was on chromosome 3, but basically no one had identified the gene. I won't go into how we found it exactly, but we did identify it. It's a BHLH transcription factor, which is kind of like a usual. So we might expect for a developmental thing, something involving hormones or transcription factors. BHLH fits the profile for sure. It took us a very long time to really prove it. So again, this is the rootless 1 mutant in our hands, and this is what we call this RT1-1, the original mutant. And of course, we couldn't use normal like Sanger sequencing and primers because you make a primer using B73 reference that has nothing to do with this 1930s landrace. So it took us a long time to find these transposon insertions here. We had to use nanopore sequencing. So we basically got this long sequence across the promoter and found all these transposons, and then we're able to do regular sequencing. So this is basically a complete knockout. So that's not very useful, but we have found 2 ACDS insertions in the maze ACDS libraries that provide phenotypes that are a little more interesting. And the main one to focus on is this rootless 1-2, which actually, you can't quite see it here. I'll show you data. It makes more than the number of nodal roots around the soil line, which is here. And then it just stops making them, unlike T43, which is basically W22 wild type. And RT1-3 is just a milder version of that. But it's this RT1-2 that I want to focus on. And so that has actually a mutation in an exon. So it's competent, obviously, to make nodal roots, but for some reason it stops making them, and here it makes too many. So there's a lot of interesting molecular genetics that we have yet to learn, and I won't talk about today, but just from the more practical, like applied standpoint, here's how we think it might be useful or what we've learned so far. So first of all, here's what it looks like. So you get these like weird, we call them bursting. It's not a very technical term, but it kind of makes sense. So instead of getting these big fat ones, you get many, many little ones often that come out of the nodes here. And so effectively what you do is this is the soil line. So these are the different nodes, which are the base of each leaf, right? And that's where these nodal roots arise. And so you basically, in the rootless 1-2, you get too many, and then it starts to go down. And this is a little bit environmentally independent, and we've done this 4 years in the field. So some years which node it shifts and the relative differences, but we always see the same pattern. In the greenhouse, it's very stark. So if you keep it in really nice conditions, it just stops making them at about node 3, and there's a huge difference at the other nodes. In the field, it's more labile. And then the phenotype is sort of enhanced actually in low end. So there's also some developmental interaction there. So the idea was like, I'll show one more piece of data. So, as we expected to have these extra nodal roots, like at deeper, earlier nodes near the soil, we expected to have more roots at deeper depths. And this is, 1 year of data. We'll have another year this year. So this, take it with a grain of salt. But we certainly were encouraged seeing this signal, especially in this region, that it did look like there are more deeper roots below basically 30 centimeters here. So we'll have more data at actually 2 different field sites from this upcoming year. So next year when I come, I'll tell you all about that. But the idea here was that it should have an effect on efficient nitrogen capture. And so in what's typically prescribed in our region in St. Charles, Missouri, 150 pounds for, I think, probably 175 or 200 bushel over an acre corn, something like that. We actually saw a benefit in that treatment. But then if you cut that in half, obviously we see a reduction in the wild type, but the RT1-2 was actually was able to capture. Well, we have other data. It captured more in, and also the yield actually matched basically. So the keen eye, and I can't even look at Tony in the eyes right now, might be looking at the yield per plant and saying, yeah.
Is that root mass? Grain mass?
No, I'm sorry. Yeah, I just realized that's super confusing. This is grain mass, grain weight per plant, and it's pretty low. because this is in W22 inbred, which is a really crappy inbred in Missouri. So Alex has been crossing this into as many different backgrounds as possible, and we would love to get this in elite, germplasm someday. I haven't figured out how to do that yet. For all I know, this gene has already been selected for in that germplasm. I don't know that either. That would be awesome. But I'm trying to figure out how to make that happen. But meanwhile, we do have this in many backgrounds, and probably next year we'll have the data to show like what it means in different backgrounds and hybrids as well. Okay, but at least in this experiment, we do see basically the steep, cheap, and deep phenotype mirrored in some way, and then we see the expected effect. We see the signal there as well. So that seems encouraging. Okay, so I'll leave it at that. I won't make any claims, really beyond that. Potential is carrying a lot of water there. Okay. But another really interesting aspect to this is if we take rootless and we look across sequenced maze lines, and here I'm just showing you what the NAM founders, which are these 26 lines that are kind of meant to encompass maze genetic diversity, or at least were 10 years ago when they were developed. And we also incorporated some other lines. So this is a line that has some teosinte integressions. This also has some teosinte integressions, the C732, which is Sherry Flint Garcia's. And so these both have these lines have more than the number of nodal roots. These lines were Z30 and Gaspé Flint, which are two totally separate. This is a teosinte. This is this weird little inbred maze line. They have the exact same PIF harbinger transposons here, and they both have low nodal roots. So we're trying to associate promoter variation, with nodal root phenotype. Like, is this just a weird thing in this just a few backgrounds, or is this maybe a genetic regulator, a tuning knob of nodal rooting? So, and then it really got interesting when we looked at the NAM founders. So this is just total crown root number from an experiment we did, and all the ones in orange, these are obviously the lowest, and 4 of the lowest 6 have the exact same hat transposon right and upstream of the start site here. And then basically gene expression is correlated with this. So we're pretty interested to continue to try to understand this gene and what role it may or may not have in like nodal rooting number, which we think is an important like phenotype with functional implications. So that's the idea with that story. Oh, and then sorry, I forgot I added a little bit of data. This is pretty new, but this is the same. These are the nodal root counts. So again, this is the soil line, just like I showed you with RT1. This is B73. So you can see it makes a lot of above ground nodal roots and fewer below ground. Different genotypes, like these are from Mexico. These are CMLs. These tend to invest in different ways into nodal roots below ground. So there's some variation there as well in these NAMs. We don't know this is tied to rootless, but that's kind of what we're investigating. And this is hard to see, but it's mostly just to show you that we're doing it. We did it, so I'll have more data coming out soon for the NAM founders. But under high and low end, this is just rooting depth. So this is down to 1 meter, and this is total amount of root length at different depths. And so we can see different patterns across the NAMs, and then interactions with nitrogen. For the most part, plants invest more carbon into their root systems under low nitrogen. That's sort of well known. We see that, but there is genetic variation for this environmental response, right? So we can see some big differences here. I mean, this could be noise, but in this case, actually, there's at least one case here, where there's like fewer. That's probably noise, but in some cases where there's even like fewer roots. This Illinois hybrid inbred here. So more on that to follow. We have a bunch of data coming out of the field this year. We just collected it all. Got a wash soil cores. We'll be doing that all winter. So come the spring, maybe the maze meeting, I'll be able to tell you more about this. Okay, so that's the rootless 1 story. The next one comes from also a really interesting old genetics story. It's ideal root architecture 1. And so this Illinois long-term selection experiment is still ongoing, to my knowledge. It started in 1896 in Illinois, and it was a recurrent selection for seed protein content, so either high or low. You plant the highest out again, you plant the lowest out again. There's also a concurrent oil experiment, but we're focused on the protein because that's tied to nitrogen, like pretty closely. So over time, seed protein concentration is like huge, right? For IHP, and it bottomed out around 4%, which you don't germinate, you don't have more than that, basically, in ILP. So the idea was, to what extent does, have changes, indirect selection for roots happen that allow these to capture more and potentially, right? That was actually a postdoc fellowship I had many years ago at USDA, and we've still been working on it. So around cycle 70, this recombinant inbred mapping population was generated as a cross between these two populations, so we can then map genes. So, and again, there's some other data that was already published just on N uptake efficiency. So as you add more and more N, IHP is very good at sucking it all up, like perfect, almost uncannily good. And then IHP or ILP at some point around 100 just basically can't really make use of additional nitrogen that's added to it. This is in a field experiment. And then, this as part of my postdoc, I could see differences in the root system architecture. But what we'd like to know is what genes are involved in this, okay, so we can actually use it directly. So we did a big field experiment across many years, dug these root crowns with shovels, did the evaluation. This is before we were doing the 3D X-ray, so we used a two-dimensional method, which is really handy. I highly recommend if you want to do root work, I would go directly to dirt and basically just setting up a little formal portrait. We do this in the field in a trailer, and we evaluated thousands of roots. And like in a week, you can dig, wash, and get phenotypes, like good phenotypes on thousands of roots. So it's been really effective for us. Of course, it doesn't tell you what's happening here. For that, you need Brazil. This worked for us. We did a big GWAS on it. We found a few interesting things, but the most interesting thing was this gene. Blank for now. I do a root architecture. So Michelle's a grad student has been working on it in the last several years. Same story here, because it was in these old land races. We couldn't forever get the lesion, but eventually nanopore sequencing came through again, and we found this huge, almost 5 kb indel, and then a smaller one here that we could then use to genotype. So then we can take each of the recombinant inbred lines in this population and say, do you have the low protein allele, and do you have this deletion or not? Okay, so we could then genotype that. So yeah, and this affected expression. So you get about half the expression if you've got these mutations. So that's kind of what we're looking at here. The gene itself, which I can't tell you exactly what it is yet. I plan to present this at PAG, so I can tell you then, but we're working on IP stuff. And anyway, but it's in central carbon-nitrogen metabolism, which is really interesting to us. Again, it's not a transcription factor. It's not directly related to hormones. It's basically involved in the plant sensing carbon and nitrogen, and then like how it makes like amino acids and what it does with those amino acids. So that was pretty interesting to us because we found it from a root phenotypic screen, but whatever those root phenotypes are clearly some like, I don't want to call them a byproduct, but they're an outcome of probably something to do with internal nitrogen sensing, right? And that affects the root structure, and then that affects how the plant actually performs in the field, especially across multiple nitrogen levels. So it's a pretty interesting thing to work on. I should have paid more attention during my plant biochemistry. I'm realizing that now, but anyways, Michelle's learning a lot about central metabolism. So same thing, we had a really nice pandemic field year. We got to see each other. That was super nice, socially distanced, basically 6 feet. So we basically planted this out, 130 inbreds. So just keep in mind, this is a mixed population. So the genetic background is heterogeneous in all of these, right? The only thing we kept that we knew was that about half had the IHP or ILP or RA1. Everything else is mixed up. So if we saw any signal for root system architecture, I thought that would be quite encouraging, and that would be indication that this gene is the actual causative gene. So corn grew pretty well that year. And we did see a phenotype. This solidity phenotype is a ratio of volume to convex volume. That tells us basically how thoroughly the plant is exploring the volume of space that it's taking up. So if you have big gaps in between, you have a low solidity, you can explore a big region and leave some unexplored, that's a low solidity, or you can grow smaller and fill that in and explore it really thoroughly, that's a high solidity. Or you could grow big and have a lot of roots, but then you have to have a big root system. So that's the idea there. So we corroborated this in 2022. Always field work is tough, as many of you know, for grad students, because stuff happens. We had a flood in 2021, but finally we confirmed this in 2022. And meanwhile, I've been working with Bing Yang at Mizzou, also joint appointment at Danforth Center, to develop a CRISPR line in V73, which we've confirmed now. So now we can actually study like the effects of this gene in a homogeneous background. So again, there's a lot more to learn about this, and we're just getting started. But stay tuned if you're interested in this space. Okay, so those are the two genetic stories I wanted to present to you, and then there's a lot I won't be able to talk about in terms of some of our other field work, but just that, as I alluded to earlier, a lot of the functional implications of like deeper roots, for example, for carbon sequestration, for nitrogen dynamics, haven't really been explored because we didn't have a good way to just make deeper roots. I mean, we compare different species, but there's so many differences, it's hard to understand what that means. Here we can basically say same genotype, different root structure because of mutations in one or a few genes. What does that mean for soil? Not just in 1 year, but year after year. So we've had long-term interest in work in AMF fungi that I won't have time to tell you about today, but we do have a hypothesis that deeper roots will allow AMF colonization deeper into the soil. So AMF are not thought to be super deep in the soil. So is that because there's no roots for them to live on, or is that because they can't live there for some reason? I mean, I don't have an answer, but we do expect roots to drive more AMF colonization deeper in the soil unless we reach some limit where AMF can't grow. And that has implications for soil health, for carbon sequestration, and just overall how plants use their carbon. So we hope to learn more about that as well soon in these multi-year experiments. And our approach is basically, since there's not one good method, we use a bunch of things. So I didn't talk much about these root trackers, which basically can give you temporal, like root growth metrics just around the root crown. Brazil's root front velocity we've used because we've been partnering with Ceterius, Arctulis, and Tony on this. And so we've learned to use this pretty effectively. Root crown excavations, obviously soil cores. And then of course we measure as much as we can above ground as well. And something also I won't have the chance to talk about today was X-ray imaging of soil macro and microstructure, because we've got these X-ray systems and we can really start to map, in particular, the pore structure and the topology of the pore structure. And for example, if we compare like a native prairie soil to a conventionally managed agricultural soil, huge differences in the amount of pore volume, huge differences in how they're connected and the sort of like how evenly the connections are. So in the conventional ones are easy to like collapse and separate the topology. The native prairie are more robust. That's kind of the take home from this work. So that's the basic approach that we take in the lab for the genetics I talked to you about, and also pretty much all the root work that we're doing. Okay, switching gears a little bit here. This is another like phenotyping platform that we've developed in the lab. It was really meant, in a sense, out of the frustration for not being able to capture freely grown 3D root systems. You can't do it in the field, even if you dig a trench and you very carefully go and try to reconstruct. You really can't do it, and even if you do, it's your life's work, and it's an N of 1, and so what can you do with that? But then, of course, you grow these things in pots, which we do all the time. Either you grow them as seedling, or the root is shaped like the pot, and there's some real limitations in that. So we just wanted to know, like, what does a freely grown root look like? And that has implications for, like, okay, I go to a field, I take a soil core out. How much noise is in that sample? Like, what is the real heterogeneity of the 3D structure of a root? And this is one way to get at that. So I think you get the idea. We built gigantic boxes, and then we just put a frame inside that holds the roots up so that we can open them up, remove the soil, and see the roots. And this is a switchgrass root system made by photogrammetry. So I'll give you a few details on like how that works. It's published now as well. So built a big box, put a frame inside. We can measure what comes out the bottom. So the idea is also a system that allows us to measure basically what goes in, what comes out, what's in plants, what's in soil, what's in microbes. Okay, so we just want to be able to track carbon and nitrogen dynamics across this system. So we use a lot of 13C and 15N isotope work to do that. So, nothing very sophisticated. And then we just use fishing line inside, and this basically holds the roots up. Okay, then we can just water it from the top. It's nice. We can water as much as we want. We can put whatever nutrients you want in it. And then the other thing that we did was we basically embedded sensors in it. So these are matrix water potential sensors that give you, the matrix water potential at this point. And they also give you temperature. So we do see a temperature gradient here. And the matrix water potential is like a physiological measure of like how hard it is for the plant to take water up, to suck the water up. And then we also have these little tubes here, a little hard to see, but we hook these up to a Picaro mass spec to measure CO2, to measure root respiration at different depths. Okay, and then over time, we basically can integrate this, and we have like a 4-dimensional like system. Sampling system. So in the end, this is what you get. This is a DOE switchgrass project, and so these are an upland and a lowland switchgrass are obviously adapted to very different environments, and you see they have like a natively very different foraging strategy, so to speak. They explore the soil very differently, and I mean, I assume, and I think it's a decent assumption that that's due to their adaptation to where they normally grow. So you can see this difference. We can also get differences like this extreme by just changing the moisture level. So I mean, there's really obviously, as I think is well known, a lot of plasticity for these environmental measures too. But the question is like, how do we turn this into data? It looks cool. We put it at the St. Louis Science Museum so kids can touch it. Looks really neat. But okay, what do we do with it? It's too big to put in our X-ray system or most X-ray systems, unless you're like Boeing. So we used a different method. This is photogrammetry, structure for motion, which is obviously commonly used in remote sensing and above ground data. But we used it instead for basically took a handheld camera, Tyler on the right there, took a lot of images. We built this like special shed to get a lot of random patterns in the background. That allowed a big GPU to align everything. And then from that, we get a point cloud. Now the point cloud we get has the room, it's got the frame, it's got everything. So we had to use a bunch of, basically image processing to remove that. But at the end, we have a gigantic point cloud of a real root system that basically was not constrained by the pot, which was pretty neat. And so again, those differences we can see by eye. We now have a digital version of those, and now we can use all the same tools that we use for our X-ray systems or our optical gel system, which I didn't explain to you. But it's a 3D data set. We can use all the same measures, so that's really nice. We're not measuring like different root systems that came from different, like we're measuring different systems or came from different sources in different ways. We can measure 3D to 3D. This is just, instead of a root crown, that's the whole root system. And so, no big surprise here, but sorghum, maize, and switchgrass, has different root system architectures. And we can actually measure some aspects of this, and there's some artifacts here. You may be seeing these shoulders. This is, because the roots slumped down on this. So it's not a perfect system. It's just one in the arsenal that allows us to some new insights. The other nice thing about it is we can open the sides of the box and actually like go in and take pieces of root. So we've done this, for example, treating like the seed with a microbial cocktail, and then saying, well, if I measure roots here, 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 and here, what microbes from this cocktail stuck on those roots? So colonization and persistence questions we can get at, right? One of the reasons these cocktails may not work is because you put them on there and they work in theory and the biology works, but they just get out-competed. They don't stick on the roots, so they don't actually have the effect that we wish they have. That's an idea, at least. It's sort of, in a way, testable in this system. Okay, and then this part we have not gotten to. I love ideas for how to do this, but we've got this endpoint 3D root system, and then we've got this like 4-dimensional. So we took the point sensors and imputed a 3D map, and then if you run that over time, you can see the fluxes in temperature, matrix water potential, carbon dioxide, or respiration. And so sort of relating the root structural information to some of this temporal information. I mean, it seemed like a good idea at the time, but actually, I don't exactly know how we're using it, to be honest with you. I'll just be really honest. So still thinking about that. But we do have some data sets. There's one more vignette, but I don't know. We may be running out of time. Should I keep going? I don't know.
About 10 minutes.
10 minutes? Okay. I don't leave time for questions, but okay. I'll keep going. Get up and leave if you want. It's okay. I don't want to offend me. So, okay. I just wanted to get this additional part, which is like this cover crop system. We've really taken an interest over the last 3 years or so in cover crops because it seems like a really impactful way to affect sustainable ag and carbon sequestration and soil health, just because they're not widely adopted. And so maybe if we can make better cover crops, reduce some barriers to adoption for cover cropping, they could be put into use. That seems like a very direct, Tony, I know we've talked about this, but you've heard this before 30 years ago from someone just like me, but we're trying it again with new tools. But anyways, we are doing this, so we'll see. Time will tell. So one interesting cover crop we've been working with is pennycress, right? Which you may have heard of. There's some buzz. Bayer just acquired covercress, which is which is the main company turning this weed, basically agricultural weed that grows everywhere, into a cash cover crop. So the idea is it's a winter oil seed. I think there's probably diploid winter annual. It's really nice because it's basically a Arabidopsis. It's basically a useful Arabidopsis. It's same genome. You can floral dip transform it, so we can do CRISPR editing. So there's so much like easy work we can do in it. It's so refreshing compared to like corn, which is real hard. It's only partially domesticated, and so this is pretty interesting because we can take all this natural variation and like leverage it towards improving root systems, improving ecosystem service function, that sort of thing. So we can start breeding efforts with both roots and shoots in mind, not just the shoots part, right? Okay, so we've had this project for a while, and we wanted a way we do field work with it as well, but we wanted a way to evaluate it at a large scale. So we went back to this large format mesocosm system, and we built like a swimming pool sized one, basically, like a huge one. And then put a little farm bot on top to measure rosette. So this thing just is automatically measuring. So there's, I think, 700 pennycress plants in here, something like that. So it's nice. I mean, we could kind of set it up like a little mini set of field plots, measure the rosettes, and so then we've got the image analysis down pretty well. So we can see variation for rosette leaf area. This is, I don't actually know which time point this is, sorry, but it's some time point where the rosettes were reasonably non-mingled and we didn't have flowering, I guess. So of course we can see genetic variation, phenotypic variation across genetics, which is great for that. And then, eventually these things get a little too big. This is no longer useful at this point. But what we did was go in and inject some 15N nitrogen at the base of each plant and basically ask, how much N wound up in the shoot. So we cut the shoots out, we ground them, measured the 15N. So we're starting to get a functional readout of like the root system differences. And so there's still a lot of root analysis to do, but we get a lot of variation for shoot 15N here. And when we correlate that with just root dry biomass, we get a nice correlation, but we also see lots of things that have high N but don't have the biggest root systems. And so we could reasonably at least hypothesize that root system architecture was involved in some of these data. So that's kind of where we're at with this, still chewing on it, but it's kind of a neat way to use the mesocosms, and I think could be pretty fruitful in this project. So in that, just continuing on the cover crop theme for another few minutes. Again, I kind of talked out of turn here, but U.S. grain belt in the summer, everyone's very familiar with this. Just an incredible thing to look at. Hopefully I'm not misrepresenting this too much, but anyways, in the winter, this is often what it looks like, right? Nothing's planted. We do get ponding, flooding. This is problematic, right, for nitrogen.
Especially in Missouri.
Especially Missouri, which is trash state, right? Not in Indiana. This definitely came from Missouri, my backyard. Yeah, but I mean, suffice it to say, like a principle of soil health, according to Soil Health Institute, is living roots in the system at all times. Obviously, erosion, excess nutrient capture, things like that. Cover crops make sense on some level. Now, practically, whether they do or not is a whole nother ball of wax. But I would like to use our science, use our plant science to try to at least address some of the sticking points. That's the big idea here. And some of it's just learning the different cover crops, how they interact with the corn. So that's sort of what we're trying to do. Obviously, there's a whole slew of ecosystem services. The nice thing about cover crops in one sense is we have a whole bunch of species to work with. So we can really leverage plant genetic variation, which is like one of my favorite things as a plant scientist, plant genetic variation. It's horrible because it's hard to work with 30 species. It's hard enough to work with corn, and there's still a lot I don't know about corn, let alone all the grasses, all the brassicas, all the legumes. But anyways, again, hard challenge is not a reason to not do it. It's actually a reason to do it. So that's the idea. And, there's awesome variation both above and below ground. Hopefully we can leverage this in some way. That's the idea. So we've done some preliminary work. And what we realize is that in the literature, there's obviously a lot of work on cover crops. A lot of it's agronomic. Some of it is phenotypic and sort of more plant sciencey. But there really hasn't been a sort of like the knowledge base of above and below ground, like across many species of cover crops, that knowledge base of above and below ground phenotypes doesn't really exist. There's patches of it, but it's hard to put together. There's a lot of contradictory work. So what we set out to do is try to basically get that, like just develop that knowledge base, at least for our farm at Danforth Center, measure as much above and below ground traits in the variation and use that to start to generate hypotheses that we'll test. So we've already done a preliminary study here, which hopefully will get published soon. We've expanded that now to about a 13-acre experiment. So this is these are 60 by 60 foot plots. They're triplicated, and there's 30 different species plus 10 mixtures in here. So it's, and then these two strips here are conventional ag, so no cover crops and tilled, and we're planting these no-till with corn. And the idea is to grow them same species or mixtures in the same places, right? GPS located year after year after year to measure changes to the cover crop, root and shoot traits, the soil, and impacts on the corn and the corn phenotype. So here's the list. There's not really time to talk about them, but we picked them as best as we could. Some of these do winter kill, but most of them don't. And we made mixtures in kind of a methodical way to test different interactions here. There's a lot of space, parameter space here. We had to pick some. And 13 acres is a lot to pull soil samples and pull root samples from. So this is sort of the best we can do. And we expect this to change over time, but this is kind of our starting place. Okay. And then, eventually, again, as I mentioned, we're very interested in sort of root traits, how the microbial community and soil properties are changing. And this is both for cover crops and the cash crop. So, again, I think this is my last slide. You can already see after we started this experiment at an old farm, we got a nice new farm, so we moved it. This is corn. The first year after the cover crops, and here's what cover crop legacy was. So crimson clover, cereal rye, ryegrass. So anyways, you can see after 1 year, and this is conventional, a lot of variation in how green the corn is, how tall the corn is, what its flowering time is. We also measured roots. So cereal rye really, in this experiment, impeded root growth. So roots grew deeper from root frontal velocity data much more slowly. No surprise. So we don't have the yield data. We just pulled these 2-3 weeks ago. But of course, we expect there to see that yield gap in full effect here. The question is why? It's not. There's some reason for it. And it's biological. There's some interaction, whether it's stealing nitrogen, whether it's a chemical legacy, it's changing the microbiome, whether the soil structure is something that's sort of is influencing it. We hope to learn here. So that's the big idea. And yeah, we're measuring all that stuff that I was referring to in terms of roots, but also as much of the soil as we can. I'm not a soil scientist, so I'm really just trying to learn as rapidly as possible what are the most important traits that we can and should be measuring. So a lot to learn here, but we're pretty invested. Hopefully you see some hope in and some of the tools that we have to bring to the table here being useful for this important problem. Okay, so again, thank you so much for your attention. I'm sorry if I went over. I really appreciate your interest, and thank the funding sources. It's a big lab. It's for really interesting group. I mean, computer scientists, mathematicians, physiologists, geneticists, field people, and so it takes a lot to learn the same language and to have common goals. But you know, we can do some pretty interesting science in that way. And obviously, I'm sure there's a lot of parallels to what's happening here with the phenomics efforts, in particular digital agriculture here. So with that, thanks again. Time for questions. Happy to take them. Okay, that's it. Yep.
Question back here.
Well, there's no chat. There's no one online, so there's no chat questions. Yeah. Please speak up too, because I'm slightly deaf. I have hearing aids now, but they're a little iffy.
I'll talk loud.
Thank you.
The selection experiment's really interesting.
Yeah.
Have you done any work with modern germplasm to see how the root architecture has evolved over time? And then also, it's pretty obvious you don't mind being opinionated if you've got ideal root structure.
Yeah.
Yeah. What's your best guess for what the ideal root structure looks like going forward? Is it steep, cheap, and deep, or do you think it's going to be different?
I think it's going to be different. So ideal root architecture was coined by a previous postdoc, and you know, we didn't think deeply about it. So I see it as a placeholder. There's no such thing as a platonic ideal of a root system architecture. The ideal root system architecture is entirely contextual on the environment and the management and what you're looking to get out of it. Okay, you know, so the one that maximizes ecosystem services or efficient nitrogen capture may not be the same one that maximizes, you know, something else. I don't know, protein content of the seed. Okay. Your first question, yeah, this is a project with Tony and Raziel and Satirius. We've been evaluating the Bayer era hybrids. This is a really large project. I just could not figure out how to fit it into the talk today. I figured you might hear some of it from Tony and Raziel over time. So if I understood your question correctly, yeah, we have basically measured in great detail. And our part is the root crowns, but you know, Satirius and Raziel have been working on rooting depth and root front velocity, and then all of the agronomic traits that you might want to measure about it and how those have changed over time, genetic gain. So actually, if you're interested in that, you're coming to the Tri-Societies meeting Monday, all day Monday, basically. The results from that project are being presented. So it's really nice. It's 40 years of Bayer era hybrids, basically every 5 to 10 years. 10 years, I guess, every 10 years. And do I have that right, Tony?
Well, there's 5 hybrids within each 5-year increment.
5-year, yeah, 5 hybrids of 5-year increments, yeah.
Basically, that's why we get
So several hybrids across
40
5-year increments across 4 years. Yeah. And these have been evaluated at, you know, over 100 sites for some traits, the intensive phenotyping with roots and everything at basically 3 areas, including here in Purdue, West Lafayette area, Iowa State area, and St. Louis area. So a lot of really interesting data there that I didn't show you today, but it's all on the way.
First, excellent talk. So one question, in the promoter region of that rootless gene, right?
Yeah.
You observe the variation of transporting insertion.
Yeah.
And expression level of the gene.
Yeah.
Among the named parental lines.
That's right. Yes.
They all have good. But compared with that mutant, the 1930-year-old mutant, what's the custom mutation you think? Still transporting insertion or something else to make that big phenotypic change?
It's actually multiple transposon. It like wiped out the 5' N terminus of the gene. Yeah.
In that mutant.
Yeah. Exactly. It's totally wiped out. Yeah. So it's a complete knockout. There are still a couple of nodal roots there is an RT2, but we have not really, we don't have evidence that it's like compensating or anything. I mean, it's a really interesting sort of developmental biology story. I'd love to get around to working on that, but we're busy with, now that we're doing cover crops, we basically, it's year-round field work, so there's some trade-offs. Yeah, so it was a total knockout. So the idea is you want to turn it down or up, but not to zero, for sure, because then you just don't have a viable plan, really. Yeah, sorry.
Thank you, great presentation. I really enjoyed all the different methods that you used.
You got the fire hose, yes.
Yeah, so I have a couple of questions, but maybe I'll just ask you one for now.
Okay, I can stick around too.
Yeah. Have you observed such a thing as too many roots for self-competition in your analysis of root architecture?
Yeah, that's a great question. I personally can't imagine how a root crown as dense as the ones we tend to see can be like useful. I mean, that can't really be, I don't think you really need that much root. Especially since the way that we actually apply the nitrogen. I mean, maybe the way that we fertilize was to squirt a little nitrogen, once a week right at the root crown, that would be great. That's not how it works, right? So it does seem like kind of a way. I mean, on one hand, it must be the case that you have a density of roots there because the origin of all the roots goes there. So you can't have roots. I mean, you can have a thin one and a lot of proliferation down here, I guess, but like, eventually, everything kind of just geometrically comes back to that point. But yeah, the number of lateral roots and things you see on the nodal roots right at that soil surface is like, but I don't have any evidence that it's like maladaptive or anything, but that's a really interesting question. And then I also suspect it's contextual, right? So if we're applying fertilizer in one way, it might be more valuable. If that was out in the wild, like teosinte doesn't look like that. I can tell you that. If you go back and look at teosinte, it doesn't invest in this super dense root crown. It has much deeper root system. It goes much wider, and there's just fewer lateral roots. So, yeah. So I don't know. Interesting science there for sure, but we haven't really approached it. Okay, Basu?
Yeah. Is mostly my opinion or my view on the ideas of Jonathan, the Steve and Dave, all those things. So I think for the audience, it would be important to know that when we talk about those things, it would be for very unfavorable conditions. Because if we do breeding in roots here in the Midwest, I don't think that concept will apply because you know that here in the Midwest we are able to travel pretty shallow.
Yeah.
So we need to define how deep the root will be.
Yeah.
So I'm thinking just about this idea, like if we talk about deep root here, so how deep would be? So that would be mostly for soils that they are not actually good.
Yeah.
So it's for low nutrients and water.
No, I mean, yeah, look, that's a great point. And, to the previous point of like, deep roots aren't good they're not always good. It depends. You may want more shallow roots. Maybe the water table is the primary factor there. But it also depends on what the goal is, right? Let's just imagine we ever did have a carbon market and there was some value out of deep roots and stabilizing, like Paustian's work, right? Let's say that was a real thing. Then, maybe it would be valuable, right, to a grower to grow deep roots, even if it wasn't like the best possible agronomic decision for yield or corn productivity. You're co-optimizing a couple of objectives here. So, yeah, I don't know. Our goal is to at least identify the genes, genetic variation, mechanisms that can be used to tune or develop different idiotypes. And what those idiotypes are is interesting to think about, but is almost...
Yeah, I brought that up because...
we're thinking more broadly than just the very specific, like corn in Indiana, corn in Missouri, corn in Iowa.
Right. Yeah, many times discussing with Jonathan Mitch or those.
Sure.
Those
yeah.
we got to the conclusion that that idea of that ideal type would be for very low output soils.
Yeah.
But for here in the Midwest, I don't think we need to think about
Yeah,
that
yeah.
idea. Because
Yeah.
here, in my experience, you can get the water table like 2 feet, 3 feet, in some places.
That's enough.
In the
Yeah.
depend where in the Midwest, but
Yeah.
we will have always that problem.
Yeah.
So I don't think talking about deep root.
Yeah, that makes
Maybe
total sense to me.
You are in California or somewhere else that there are more dry conditions. Probably
Yeah.
there you will
In the great corn belt of California. Who knows? Maybe we'll live long enough to see that.
Yeah, I just wanted to point.
Yeah,
that
no,
out.
that's an excellent point, and I fully agree. My agenda isn't deep roots per se, but that's kind of what the target we had in terms of finding genetics that work for that. We made some progress there. Okay, yeah, now how are we actually going to use it? That's a second level question maybe
Yeah,
for me, but that's a good to
Nice
keep in
talk,
mind.
by the way.
Yeah,
Chris, first of all, thank you for your talk. I have a question about the deep roots, soil carbon, and yield. So we know that the modern corn yield has been increasing steadily.
yes.
2%,
Yes, I've
3%
learned
in
this.
the last
Yes,
30,
yes,
40 years, right? We hope to continue to see that increase.
yes.
If we breed or choose corn varieties that can have deeper roots
Yeah,
to increase carbon sequestration,
yeah,
well, we compromise the yield increase.
yeah.
And we have the balance
Yeah,
above ground, below ground.
yeah, yeah. I mean, in the absence of an economic incentive to not, maximize yield, it's a non-starter. If any of this root genetic stuff affects yield in a negative way, I realize that it won't be used. So my goal in terms of the applied work is for it to be used. So it depends where you grow it, because basically, good investments can yield a bigger return. Poor investments can do the opposite. So investing in roots, it's not a hard trade-off. There's certainly no genetic evidence of it. There's scientific evidence in certain situations, but it's not like if you invest in roots, that always has a negative effect on the shoot. If you invest in roots and it provides a functional benefit, capturing more nitrogen, avoiding drought, stress, this has definitely been shown in rice, that has a very positive effect on yield. Maybe in the Midwest with a high water table under our modern agronomic conditions, investing in roots actually does have this negative effect. That is certainly possible, but again, depends what the objectives are, and depends on what the sort of the production environment is, I guess, in the case of corn. Yeah. Next. Yeah, Mitch. Yeah.
So I'm just sitting here reflecting a little bit on your work. And you talked a lot about what the plants are doing. I was just wondering if you could say anything you didn't talk about this, and maybe this isn't exactly your area of science. You talked a little bit about arbuscular mycorrhizae and some of the other creatures that plants talk to.
Yeah.
Can you talk a little bit about opportunities for harnessing the broader, whatever you want to call it, microbiome, microbiome, or the complex underground ecosystem where these plants live, and what the opportunities might be to do genetics in that space?
Yeah, great question. I guess I'll start with my basic perspective is, obviously plant breeding works, but the strong selective pressures, are conditioned by the management practices that we breed under. Okay, so I think you can make a really plausible argument that we have been disincentivizing, for example, modern corn to make productive alliances with AMF fungi and even end-fixing microbes because they don't need to, right? So I think, certainly evaluating exotic germplasm and exotic alleles that may not be in the modern breeding germplasm. So we've done a lot of work with teosinte synthetics, which are Sherry Flint Garcia's population. She crossed all the NAMs plus Zea parviglumis in. So the idea is to bring in genetic variation. Some of that genetic variation might be influential for more productive interactions with AMF fungi or other microbes that have other benefits. So again, this would involve eventually growing corn in a different way, which is maybe not very likely, but in terms of certainly the genetics and understanding those interactions and trying to, I guess, again, my perspective is like we want to tilt it in the favor of the plant, right? We want to tilt the interaction in favor of the plant. Yeah, novel alleles, exotic germplasm, because, 100 years ago, we didn't have synthetic N fertilizer, and 10,000 years before that, teosinte was growing in the hillside with very little additional, and it still grows there. And so plants, of course, had to rely on fin-fixing microbes and AMF fungi to get their nitrogen. There's definitely good genetics that will be make interesting stories that we can learn mechanistic genetics, leveraging that germplasm. I don't know how we're going to use it, or if we'll be able to use it, but so.
I was just thinking of the examples of the low-end tolerant SINC germplasm that's been released and other things that,
yeah,
I know Tony's looked at that, and it's not exactly clear how any of those things work.
Oh, really? Oh,
Yeah,
okay. Oh, okay.
Tony, do you know something
Did
about
you look at the roots?
I don't know. We will need a lot more years before biological N fixation will be effective at reducing the N rate by even 30 kilograms per hectare in the Corn Belt.
Which I can't think in grams and hectares, but that's what percentage? 10%?
No, I just meant to actually accomplish
use something.
a reduction in the N rate. So let's say the N rate right now averages about 180, 190 kilograms
so
per
30
hectare of
being
N, and so basically
15%,
just
20%.
So I mean, we're still a long ways from that.
Still a long ways, and we'll
That
always
doesn't
be a
mean
long
that
ways.
it's still good science to pursue. Because if we can do that in the context of reducing N2O emissions and at the same time making improvements, rhizosphere related or whatever, then it's an objective.
Yeah.
That has other benefits, but in the short term, it's been easier to prove benefit in the greenhouse than it has in the field.
And the only reason I'm asking is when European policies come down and say this is the way it's going to be done,
right?
Then it changes.
Where do they turn? Right. Yeah. So that would obviously change the whole paradigm. I don't think we're very close to that here, but yeah, corn is grown all throughout the world.
When it's policy, it doesn't have to make sense. I'm just saying things can happen.
Yeah, that's right. It really doesn't make sense.
Thank you.
Yeah, so yeah, I mean, we're getting pretty good at synthetic biology, so I think there's some interesting, there's a lot, fungi are hard to work with, obviously, but there's been some pretty good progress in the synthetic community space. But I think, again, on principle, these things work. We know microbes fix N. We know they can give that N to plants, although they don't often want to. That's its own problem. But how are they incentivized to colonize and persist? Under what conditions do they really engage in the symbiosis? And under what conditions do they not? I think are pretty unknown. Very really interesting science questions. So yeah. I don't know if I answered your question, but we can talk all day about
I know that
this.
wasn't the topic you're talking about. I was just thinking about.
Yeah. I mean, I've been working with Valent Biosciences for many years, who has a whole rhizosphere group and soil health, and their approach is, basically finding isolates that just work better from the natural system. So we've been using some of our tools to test that. Do these things actually So like the PET work, I didn't have time to show, but do they draw more carbon down, isolate 1, 2, 3? And we do see variation for how much carbon they draw down into the root system, for example. I mean, what that means eventually for the plant, how you can control that in a field which is chock full of AMF plus every other fungus plus every other, creature under the sky. I don't have a good, it may be synthetic biology. We may need to engineer systems where they talk to each other exclusively, have to get clever about it. I don't know. Yeah. I'll be pondering it on my drive home for sure. That's what I think about, when I
Well,
can.
the tools, with the new tools, you can do things.
Yeah,
I mean, I don't
that's
know
the
if
hope.
it's better or different, just
Yeah,
different. How you
that's-
make it different.
That's the hope.
Right.
Someday we'll get a new insight that might actually be the thing that makes a change. I don't know. Thanks
Thank
so
you very
much,
much,
really, for
guys.
staying extra long. I appreciate it.